Okay, welcome to chapter 8. This chapter is about cell division. All right, so cell division is a term that we use to describe exactly what it sounds like, how cells divide, but it also is referred to as cellular reproduction. So cells can reproduce themselves, replicate themselves, or in other words, divide. So cellular reproduction and cellular division are kind of flip-flopped at times, but either way, remember from chapter 1, reproduction is a characteristic of all living things. Okay, therefore reproduction is going to be found on a cellular level. Okay, and that brings in the cell theory and also some other aspects that are necessary for life, to continue life, to maintain a physical body. All right, so if you remember, the cell theory says all living organisms are made of cells, at least one cell, and all cells come from pre-existing cells. Okay, so there's 3 forms of cell division. Binary fission is the first one. Mitosis, meiosis. Binary fission is a method used by prokaryotes. Prokaryotic cells, they reproduce this way. All right, so let's see exactly what's happening here. So this is the parent cell. Okay, I'll go ahead and label that real quick. This is a parent cell, meaning it's the original cell that it's about to be replicated, reproduced, and or divided, same thing. All right, so before this cell can divide, a couple of things need to happen. We can't just cut a cell in half. That would destroy the chromosome count. All right, so what happens is the chromosomes of this organism have to be copied. So see this little coiled up mess? Okay, of course, those are the chromosomes. It's DNA. So every single DNA nucleotide has to be replicated in its entirety. All right, so that's happening here. And when we get to step 2, or step 2, you can clearly see that there's one set of chromosomes right there, and there's the other. And on top of the duplication of chromosomes, the actual cell itself has to grow. So it's growing in length. See how long it is compared to the original parent cell? Right. All right, so it'll continue to elongate, and then it'll start to split. See how it's making this kind of inversion? It's starting to divide, and this will keep happening, and it'll get closer and closer until we have split the cell in half, and we have 2 genetically identical daughter cells. All right, now that terminology is really important. 2 genetically identical daughter cells. All right, very good. Okay, so what about eukaryotes? That's how prokaryotes reproduce. What about eukaryotes? Well, this is where mitosis and meiosis comes in. So mitosis is one type of division, meiosis is the other. Now, what's the difference? There's a massive difference. So here we go. So mitosis is designed, or what it actually does is to take our cells, or cells in general that are eukaryotic, and reproduce them. Now, why would we need that? Why would we need to generate new cells? Well, we do. We need the generation of cells for growth and development. Okay, and we need it for any kind of cell renewal and cell repair. So you have cells that divide every 24 hours, your skin cells. You have cells that get damaged and they need to be repaired. That's through mitosis. Okay, now look at this term. This is important. It says mitosis generates new diploid cells. Remember diploid? Diploid, D-I, di. Okay, diploid means that the cell has the full set of chromosomes for that species. Okay. All right. So let's do an example. Humans, for example, our full set is 46 chromosomes. Okay. And when we write it out, it would be, that means diploid. Okay, so, alrighty, meiosis. Okay, meiosis is used for one reason and one reason only, for the production of gametes. Gametes are the sex cells. Okay, so see, I want you to know this term, gametes, because I'm going to use it. But these are the sex cells, like eggs and sperm. The reproductive cells. And notice that it says meiosis generates new haploid cells. What does haploid mean? Okay, haploid means that the organism has half the number of chromosomes for that species. Example, let's say we're talking about a haploid sex cell. Okay, so I'm not going to really write anything other than this. So the count, the chromosome count should be 23. Okay, so in other words, eggs and sperm, each one is carrying 23 chromosomes. All right. So what we're looking at here, this is called the human karyotype. So scientists have figured out what our chromosomes look like. They basically figured out all of our genes, and through the Human Genome Project, they know what they do, what they encode for, and things like that. Now, what this is your human karyotype. So even though each person has unique DNA, it doesn't really matter that our DNA, well, it matters, but in regards to the human karyotype, your chromosomes look like this as well, even with your unique DNA. So what we're looking at here is a layout, a display, a physical display of what our chromosomes look like. Okay, notice that each one has a number next to it. So there's number 1 all the way to 22. And then these are called autosomes. Autosomes. Okay, now the 23rd chromosome, of course, is the 23rd set is the sex chromosomes. All right, so earlier I said that humans have 46 chromosomes, and we do. And this is what they're supposed to look like. So you really have 2 sets, or 1 set. So 2 chromosomes at each location. There's chromosome 1. That's another chromosome next to it. Okay, so you have a these are called homologous chromosomes. So you have homologous chromosome set number 1 right there. Okay, that's another chromosome. That's another. Okay, so you have 23. If you go through all of these, 23 pairs. And then, of course, if you are female, you're going to have the XX chromosomes. males have the XY, and that will determine the sex of the person, the baby, the individual. Okay, now if you count them all up, 23 sets, that's 46. All right, now another important factor here, and this should be something that you basically take home and remember permanently. You inherit 23 chromosomes from mom and 23 from dad. So one of these, we'll just say the first one, okay, is the mom chromosome. Okay. The second one is the dad chromosome. This one's maternal, paternal, maternal, paternal. So either way, you get a total of, when you inherit your chromosomes, when you're born, when you're made, right, you get 23 chromosomes from mom, and you get 23 chromosomes from your dad, giving us a total of 46 total chromosomes, right? And then also I want you to see this term as well. And really what it is we have 23 pairs of, okay, make sure you're taking notes. This is homologous chromosomes. All right, very good. Let's go ahead and move on. Now, inside of a cell, I'll come back to this picture. Now, let me just kind of recap what we just discussed. Now, this is the human life cycle. Okay, here we go. Okay, now, every single individual starts off life this way. Okay, you get 23 chromosomes from the mother. Okay, notice this says haploid gametes. That means that there's 23 chromosomes in this egg. Okay, that's the mother. So this is technically N equals 23. So they expect you to just know that it's haploid when they use this, when you see N. Okay, so there's 23 chromosomes in that egg. There's 23 in that sperm. Okay, they're going to fuse together through fertilization. And everyone begins life as a single celled diploid zygote with 46 chromosomes. So yes, it's 2n equals 46. All right, so how do you go from being this little tiny single-celled zygote to a full human adult made of trillions of cells? Through mitosis. Through mitosis. Okay, so we get from here to here in the majority of the part being mitosis. Of course, there's other factors involved in growth and development, but mitosis is key. Now, every single one of your cells, every single one except red blood cells, is carrying your full DNA that you inherited or that you acquired in this process right here. So this DNA is, okay, let's say this is you. Okay, that's your genetic makeup right there. That DNA is found in every single cell because of mitosis. Because mitosis takes cells and copies everything before it moves on. Okay, now we're going to learn how that happens, but this is important to kind of put together big picture stuff when it comes to cell division. So mitosis is used for growth, development. Okay, and then once you're an adult, you need cell renewal You need cell repair sometimes. Alrighty, so mitosis is responsible for that. Now, meiosis is for one reason and one reason only. Okay, in the female body, this happens in the ovaries, and the only purpose of meiosis is to make haploid gametes. That's all. That's the only reason for it. Now, it's a big process, but I want you to make this distinction now. Okay, in meiosis in the male body, this is the production of the haploid gamete sex cells occurs in the testes, of course, and then that is the purpose of meiosis. Meiosis has one purpose and one purpose only: to generate the sex cells, to generate haploid, generate haploid. What's wrong with me? Haploid. There we go. Generate haploid gametes. That's all. All right. Now, let's go ahead and look into a couple of important factors before we jump into the steps of mitosis. Basically, let's talk a little bit about DNA. Now, in chapter 4, I mentioned that DNA, when it's unraveled, is called chromatin, or referred to as chromatin. Now, I want you to understand the structures of chromatin and what chromosomes are, and this is a new term called sister chromatids. All right, so this shows quite a bit. All right, so here's the DNA. It's a double helix, and notice that it's uncoiled, okay? And it's wrapped around these proteins called histone proteins. So this is chromatin. All of this uncoiled stuff. There we go. Okay, now chromosomes coil up and they make that chromosome shape that we saw earlier, like this. Okay, look at them one at a time. Okay, so see this here? This is one chromosome. Okay, I'm drawing around it. That's one chromosome. All right, now when cell division is happening, it can't just happen. Remember, with the binary fission, what was the first thing that needed to happen? We had to copy all of the prokaryotic DNA. Okay, well, eukaryotes work the same way. They're going to have to copy every single chromosome that we have, and in order to reproduce a replicate cell. All right, so once it's done so, okay, once it's copied itself and basically made a complete duplicate of itself, that is called a sister chromosome, or I'm sorry, sister chromatid. It's a replicate chromosome. Okay, so in other words, every chromosome that you have is going to be copied. Okay, it's duplicated, and on top of it being duplicated, it's also held together with a protein. That protein is called the centromere. Okay, and now this is an exact replica of this, and they're held together, and now they're referred to as sister chromatids. You want to know what that term is because you're going to see it quite a bit. Okay, so for a time, the sister chromatids will be held together by a centromere, and then at some point they will just be distributed to daughter cells, to two daughter cells. All right, so that's that. Now I think we're ready to talk about the cell cycle. Okay, so the cell cycle is a cycle that every single cell has to go through in order to divide or replicate. So basically, I want you to understand this very well and pay close attention. All right, so look at this pie chart here. See all of this area? This whole thing goes all the way around. It takes up most of the cell cycle, and it takes up most of this chart. Okay, because it is the longest part of the cell cycle. It's called interphase. Okay, now the second part of the cell cycle is called the mitotic phase. See this little blue slice of the pie? That is the mitotic phase. Okay, so let's start with interphase. So if a cell is going to enter the cell cycle, let's say a cell is going to divide, it has to be instructed to do so. Cells cannot just go into the cycle and start dividing without any control, because that is the exact definition of cancer. Cancer is uncontrolled cell division. So normal, happy, healthy cells that don't have a problem, that aren't cancerous, they follow these steps. Okay, so basically, the first thing is a cell has to be instructed to enter the cell cycle with growth hormone. Okay, growth hormone does exactly what it sounds like. It is responsible for growth. Okay, and that includes cells and their replication process, right? All right, so growth hormone is released and the cell will enter the cell cycle, and it will enter the first part of the cell cycle, which is called the G1 phase. Okay, what happens during this phase? Well, the cell's going to have to grow. Okay, during the G1 phase, what's going on is that the cell is growing and in size. So the plasma membrane is growing, let's see, I'm going to add some things to this list here. Let's move this down. There we go. Okay, so G1, what's happening here? Aside from growth, this is a primary growth phase. Now, the things that are going to grow are everything, okay, except for DNA. The plasma membrane is going to grow. The cytoplasm will grow, okay, and all of the organelles, all of them, are duplicated. Okay, everything. I mean, the rough ER, the smooth ER, all of the mitochondria, the Golgi, all of these guys have to be copied, because at the end of the cell cycle, we're going to get two identical cells. How do you get two identical cells? Well, you have to copy everything, and I mean everything. All right, so all of the cytoplasm, the cell membrane, all of the organelles, proteins, etc. are going to be duplicated, okay, in G1. Now, once that cell has left the G1 phase, it will move into the next phase. Okay, the next phase is called the S phase. S stands for DNA synthesis. Okay, so everything is duplicated and copied here. Now in G1, now in the S phase, the only thing that's copied is DNA. Okay, and once DNA is copied, everything will be duplicated. All the DNA will be duplicated. So let's say we move into the next step, the G2. How much DNA do we have if this were a human cell? Okay, well, if this was a human cell, a normal human cell, right, then it should have how many chromosomes? 46. Should have 46 chromosomes. Okay, now as it enters the S phase, it will have 46 chromosomes. Okay, when it's done with the S phase, where all the DNA has been copied, every single chromosome, and it's going to be duplicated, and it's also going to be connected to its sister chromatid. Every single chromosome is copied and clipped together with a protein, and they're sitting now in sets of sister chromatids. And actually, it's more like this. All right, so this is a human karyotype after the S phase. Everything has been copied. Okay, so notice that there's no longer 2 chromosomes at each location. There's 1, 2, 3, 4. That is that. Everything has been duplicated. Notice that these 2 are connected. They're sister chromatids. That means these are sister chromatids as well. So are these, so are these, so are these, so are these. And it goes all of them. Every chromosome is copied and clipped together with its duplicate. All right, so that's a big process. That's the S phase. Now, the G2 phase is when a cell enters G2, it's going to have double the number of chromosomes. So it's going to have 92 chromosomes here. And this is the final growth phase before cell division occurs. And cell division, the G2 phase is going to check. There's going to be a check point in there where the cell is going to be checked for errors, making sure that there are no errors, that every single chromosome was copied, that every single thing occurred properly, all the organelles are duplicated. It'll grow a little bit more, and then ultimately what's going to happen here is that the cell will enter the next part, which is the mitotic phase. The mitotic phase is broken into 2 parts. Mitosis, right here, and cytokinesis. They all make up the mitotic phase. Okay, now here's the deal. Do you see how small the gap or the part of this cycle mitosis is? It's tiny. It's this little tiny sliver in this big cycle. So I want you to know the cycle completely, every single part of it, and what is happening in each phase. Now let's look a little closer at the mitotic phase. Okay, the mitotic phase is 2 parts. Now actually, really quick, let me just make this even more clear. Interphase, okay, if I asked you, how many phases does the cell cycle have? What you would tell me is it has 2 parts. It has interphase, number 1, and the mitotic phase is number 2. That is in reference to the cell cycle. Now, if you were asked, what are the phases or subphases of interphase, then you would list off G1, S, G2, and then of course you have to know what's happening in each phase. Okay, now the mitotic phase is broken into 2 parts, mitosis and cytokinesis. Okay, so it's 2 parts. Now, mitosis, what that actually means, the definition of mitosis is the nuclear division, meaning division of the nucleus. We're going to divide up the duplicated DNA and distribute them to 2 daughter nuclei. A nuclei is capable of dividing in half. So those terms right there are incredibly important. All right. Mitosis is nuclear division. Cytokinesis is when the cytoplasm divides and finally forms 2 daughter cells. 2 genetically identical daughter cells. All right, but we'll get into the details of that. Now, mitosis is this subphase of the mitotic phase, also referred to as the M phase. So sometimes the book will just call it, they'll call the mitotic phase the M, okay, the M phase for short. So if you see that, it means the same thing. All right, now mitosis is part of the mitotic phase, and the mitosis has several steps. It has prophase, something called metaphase, anaphase, and telophase. Okay, now I want you to, we're going to learn each one, but quickly, I want to emphasize something. A good way to remember these steps is like this: PMAT, PMAT, prophase, metaphase, anaphase, telophase, PMAT. Okay, and that's a pretty easy way to remember it. Okay, this is the first part of mitosis. So the book will sometimes tell you that there's 5 phases, but we have condensed them into 4. The video will tell you that too. So basically what they're going to say is that there's a prophase and that there's a pro-metaphase. So what we've done is we've taken prophase and pro-metaphase and put them into one category and just called it prophase. So all of this is happening in prophase. All right, so what we're looking at here is clearly a diagram cell. And then this is a real cell, and it's been fluorescently stained. Just show you this real quick. And it's fluorescently stained so that we could see everything. Okay, so this is the cell, and all of this blue stuff right here, these are the chromosomes, okay? And this is where the spindle is going to form on each side. All right, so if we look at the parallels of the picture along with the diagram, let's take a look at what's going on here. Okay, so there's certain things and ways to remember all of the steps. And this is how. In prophase, the following occurs. Something called the mitotic spindle is going to form. What is that? Well, that's this. See these little guys right here? These are called the centrioles. Okay, and they're not usually in that position. See them in green up here? Yeah, okay, they're not usually in that position. During prophase, they get into that position, and not only do they do that, they make this net. See this kind of, it almost looks like a net, okay? This is called the mitotic spindle, okay? And both sides are going to make it because we have 2 poles. Look at the cell like an equator. So when we say pole, we mean north and south, okay? So we have the mitotic spindle formed. The centrioles, which are these, are going to migrate to the poles of the cell. All of our chromatin is going to condense into visible chromosomes. So normally we cannot see DNA, okay, unless a cell is in the middle of cell division. Then you can see it. Clearly you can see it, and you can see it right here in this image. Okay, right there, all that DNA, you normally wouldn't be able to see it. Okay, but we're in prophase, so you can. Now, the other thing that's happening is we have a nuclear membrane that surrounds our DNA. Remember, our DNA is not floating around like a bacteria cell. Instead, no, it's encapsulated in a nucleus. So how does a mitotic spindle get its little hands on these chromosomes? Is because the nuclear membrane is fragmenting. It's disappearing. It's disintegrating. So that's something that happens in prophase. Okay, so the nuclear membrane is going to dissolve. And generally the cell will kind of slow down on any metabolic activity. Metaphase. during metaphase, now I want you to pay attention to this. This is pretty interesting. Between prophase and metaphase, we've come a long way. Now, what's happening here is the centriole and its mitotic spindly arms are connected to one of the sister chromatids on this side, right? And this one is connected to these sister chromatids on this side, okay? And that goes for all the chromosomes. Now, there's that. And they have full control over the chromosomes right now. These are technically sister chromatids. And what they're going to do is push and pull and move and align all of the sister chromatids at the equator of the cell. That's where they are. Now, if we look at this picture, which is actually a great picture, you could see it. See all the chromosomes right there aligned, and yeah, they kind of look like they're way up here, and because it's DNA, but they are aligned at the chromosome. At the equator. And then this is one centriole. You can clearly see the mitotic spindle in green. Centriole, mitotic spindle. Okay, very good. So metaphase is known for one distinction. All of the sister chromatids line up at the center of the cell or the equator of the cell. Okay, anaphase. All right, so what happens here? Pretty straightforward. This mitotic spindle was not always there, right? So it grew out of the centrioles, and in turn it can retract or shorten and move this way. Okay, now if both sides, the North Pole centriole and the South Pole centriole, their mitotic spindle shortens on both ends, it's going to pull the sister chromatids apart. And that's what's happening in anaphase. And what's cool is you can see that happening right here. See how they're pulled apart? And look at this, the mitotic spindle shortened. Perfect. All right, so anaphase is when sister chromatids are pulled apart and or separated. And then on top of that, now they're going to move towards the poles of the cell. So they're going to move to opposite ends of the cell. One will move to this side, okay? And one set will all move to this side. So I guess we can grab all of these. They're all going to go this way. All right, so next, telophase. Well, now the chromosomes have been pulled apart and they've been pulled to opposite ends of the cell, and so we don't need the mitotic spindle anymore. It can disintegrate, and so it does. And then our chromosomes don't need to be in the form of chromosomes. They can uncoil and unravel and go back to chromatin, and so they do. Okay, and then we're going to need our nucleus back. We can't have DNA without nucleus for long. We can only allow it, or the cell will allow it during, the cell division process, but it's coming right back, and there it is. You can see these little fragments. The nuclear membrane is reforming around the DNA, okay, on both sides. All right. Okay, and the cell is starting to divide. See this? This is called the cleavage furrow. This kind of this whole the cell's kind of folding in on itself, and really it's not folding. What's happening is we have these little contractile rings, and they're going to keep contracting, and they're going to keep pushing and pushing and contracting until the cell, this is going to keep moving this way, and this one will keep moving this way. till they cut the cell in half. But that doesn't happen yet. We're still in telophase. All right, so that's the end of mitosis. Okay, remember PMAT, prophase, metaphase, anaphase, telophase. Now we're going to look at cytokinesis. Now cytokinesis is when it technically is going to happen simultaneously with telophase because it happens so fast, but the fact is that cleavage furrow is going to form, and those contractile rings are going to cut this cell in half. And we're going to end mitosis with 2 daughter cells, and one of the most important parts is they're 2 genetically identical daughter cells. That means the DNA here and here are exactly the same. And that is the reason why every cell in your body has the same DNA, is because of the cell cycle and because mitosis is so precise, and it typically doesn't make mistakes. Mistakes can be made though. Okay, now plants are eukaryotic, if you remember. Therefore, their cell cycle is not different than our cell cycle, right? The only difference is the last part, the cytokinesis. So when our animal cells divide, they're going to have a cleavage furrow. When plant cells divide, they're going to have a plant cell wall forming, and really it's called a cell plate for now. Okay, so basically what happens is, don't forget this is all made of cellulose, this plant cell. Let's take a look. All right, so this is all made of cellulose. It's a cell wall. So we need these vesicles to drop off this material to make the cell wall. Okay, so we have these little fragments of the cell wall, and they'll fuse into a plate temporarily until they keep growing. Okay, they'll grow this way and this way until we have a completely new cell wall and 2 genetically identical daughter cells. And that is mitosis. Okay. So, you want to know everything about interphase. Prophase and metaphase you'll put together into one. Every single thing that's happening, what's special about it, what is everything called, etc. Prophase, prometaphase, all one. Okay, then metaphase, anaphase, telophase, cytokinesis. All right, now I'm going to show you 2 animations. One is my animation that is incredibly significant. I want you to basically I want you to watch it, draw everything, take notes, print out the slideshow that matches it as well. And then we're going to have another video, and that's a great video too. So after the videos, which are mandatory, then we're going to finish the rest of the lecture. All right, let's check that out.
During the mitotic phase, a cell will undergo mitosis to form 2 new nuclei, and then divide to form 2 new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into 2 new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the 2 sets of chromosomes. Mitosis is now complete. After mitosis, 2 two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called G1, S, and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap 2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and two centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands, or spindle fibers, are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the two ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the two new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The two new cells are now ready to grow and perform their specialized functions.
All right, now, can any cell enter the cell cycle at will? What are the rules and regulations to cell development and cell growth? Okay, so there's a couple. Let's start with this one. over here, okay, to the left. Okay, this is called anchorage dependence. And there's also 3 other factors, a cell density factor and a chemical growth factor. Okay, and we've already talked a little bit about growth factor. Okay, so it's actually, yeah, here we go. So let's actually look at both sides to begin. If you take cells, okay, and you suspend them, you put them in liquid. So see all of these cells, they've been placed in this cultural cell suspension. And basically what that means is it's got all of the nutrients and factors that are required for a cell to survive. So they have all the food and nutrients that they need, right? So they can divide if they need to, but they're floating around, meaning that they're not anchored to anything. So they don't actually grow. So if we count up the cells, okay, so they've been separated here. Okay, there's no treatment. This is a control. And this is an experimental group. Alrighty, so there's that. Alright, so they put 4 cells in here. And if we count them up, there's 1, 2, 3, 4. They waited a certain amount of time, and there's only 4. Okay. Well, that's because cells cannot grow in a liquid. They have to be anchored. Okay, so they took 4 cells and placed them in a petri dish. This is technically, some kind of plastic, but there's an anchorage. They can hang on to something, and so they do, and they will grow. They will grow a perfect layer, and it's a single layer because they know when to stop. When there's enough cells in that location, they will stop. That's in reference to the cell density control. So that's that. All right, now let's look at some factors here. Okay, so anchorage is important. Now, the other things that are important. If you remove a cell, density is important. If you remove a cell or 2, they will be restored immediately. And the exact amount of cells removed, so they took out 1, 2. 2 will be replaced because the cells know that there's something missing. They can tell, they can feel one another. All right, now the other experiment. Now we started off taking these cells, right, and putting them in this liquid and in a control with nothing. They won't grow. They're not supposed to because they follow that whole anchorage dependence law. Okay, now, but what can happen is this. If we add something called growth factor is a hormone, and we add it to the experiment, to the cells, they will grow. They will grow because growth factor initiates cell division. So if you look at these 2, sorry about that, there we go. Let's look at these 2. We start off with 4 cells, and then we have, a significantly higher number. I don't want to count them all. There's something like, I don't know, we can 10, 12, 15, something like that. All right, so either way, these are the things that affect cell division, meaning that they follow laws, and there's certain things that can break the laws, like the growth factor can break this law, okay, but mostly they have to be anchored to something. And in the human body, they're going to be anchored to your underlying tissue, like they're going to be anchored to your cartilage. They're anchored to something. Okay, they're not floating around. And then the cell density is a factor. This whole perfect single layer is an important factor, and the fact that it's restorable is significant. So they don't grow out of control. They know when to stop. And then growth factor, again, has a direct effect on growth. Alrighty, so let's check out this this whole thing. So we have something called checkpoints. Now what we're looking at is the cell cycle, but we're looking at something called the cell cycle control system. The cell cycle control system. Okay, what does that mean? That means there's a system that controls the cell cycle. That means that cells cannot just enter the cell cycle whenever they want to. And they can't do whatever they want to. They have to be controlled and regulated, or we would just have pandemonium. All right, so this is how it works. First, let me get an arrow. Hang on, guys. There we go. All right, growth factor. This is a hormone. It has to bind to a receptor protein, and it does. That's the signal. This is the kind of go to the cell cycle signal. It says, okay, you're allowed to grow. Go ahead and enter the cell cycle. So it does. And it enters at G1. And now see these little, they look like lights, like traffic lights. Okay, there's a reason for that. Okay. This is where the checkpoints are. Okay, so we have a G1 checkpoint. A G1 checkpoint is going to check that the cell is supposed to really divide. So it makes sure that it does. And now, if it gives it the green light and it allows it to move through, it will usually move through the entire cycle, unless there's a problem. Okay, so there's a second checkpoint at G2, the G2 checkpoint. This checkpoint is going to make sure that everything was perfect in duplication. And that means DNA, all of the organelles, it's going to make sure that the cell is big enough. It's going to make sure that the cell is ready for division. And that is the G2 checkpoint. Okay, let's say that there was a mistake. Let's say that the G2 checkpoint caught a miscalculation of chromosomes. There's a DNA, there's an error in the DNA synthesis. Okay, the cell has to be destroyed, and it will be. So it's removed out of the cycle. Okay. And it goes into a programmed cell death state called apoptosis. Okay, so this is programmed cell death. It's like cellular suicide, because it could be, let's say there was a mistake in the gene replication, and it could be a mistake that could cause cancer. So it has to be destroyed. So our cells and our body and our system is so sophisticated and savvy that it just keeps track of everything, and it doesn't let anything through the cracks in a healthy system, right? Of course, there's things that can damage that, but that's a different issue. All right, so let's say that the cell, that's apoptosis. Let's say that the cell did well, everything went perfect, and it goes on into the M phase. There's a checkpoint here in the M phase to make sure that during anaphase, those sister chromatids were indeed divided. Because if they're not indeed divided, we're going to have a big problem on our hands, a huge problem. That's a literal genetic disorder. And and then let's say it passes, then it's done, and they'll leave the cycle and go do what it's supposed to do. Of course, if it fails the test, apoptosis it is. Okay, so those are the 3 major checkpoints. What about this one? G0. Now, G0 or G0, this is a non-dividing state. So lots of our cells just sit in this non-dividing state until instructed to do otherwise, meaning they're given some growth factor by the body and told, go ahead and, go to the cell cycle. Now, there's some cells in our body that stay in G0, which is a non-dividing state. Okay. So liver cells stay in there. They can be called back to the cycle by growth factors and things like that when there's injury, but they really don't replicate often. Liver cells don't. And that's why people that drink too much alcohol, like alcoholics, can get cirrhosis of the liver pretty quickly. And it's because our liver cells do not go through the cell cycle very often. All right, now, yes, the cirrhosis can lead to some liver cells dividing, but it's not going to repair all of them. Okay, now some specialized cells never divide, meaning after they're formed, they never will divide again. Nerve cells, muscle cells, they never divide. That means if you damage a nerve cell, you're done. It will never fix itself. That is why people get paralyzed or nerve damage is permanent, because of the fact that they do not go through mitosis. They stay in a G0 stage. Same thing with the muscle cells as well. They do not leave the G0 phase. The liver cells can, but not very often. Okay, I'm going to actually skip over this. All right, so now we just talked about mitosis, the cell cycle, and controlled cell division. Controlled cell division is very important, but uncontrolled cell division is not. Now, if there's cells that slip through the cracks, which there are, that can lead to cancer. So cells can have damaged DNA. Cells there's a range of genes that can be affected and damaged that play a role in the cell cycle and lead to uncontrolled cell division. So controlled cell division is great. We need it. Okay, but it'll stop eventually when it's done doing its job. A cell that has damage to its DNA, okay, is going to just keep dividing and dividing and dividing and dividing and dividing over and over and over and over and over again. And it won't stop. And then we end up with a tumor. Now, here's something I want you to take away from this whole thing that's significant. Everyone has unique DNA on the planet. Everyone, except for identical twins. That's a different situation, but for the most part, everyone, unless you're an identical twin, you have unique DNA. Okay, therefore, let's say a person gets cancer. See all of these cells, these cancer cells? What do they have in their nucleus? They have unique DNA, every single cell. Therefore, here's the problem. Now, let's say we have a group of people that have breast cancer. Every single breast cancer patient has unique DNA, and their cancer is technically unique. So there are, breast cancer treatments, and there's cancer treatment medications and chemo and radiation, and there's ways to destroy the cells. But what's happening is this is not a virus or a bacteria. These are the host or that person's cells with their DNA. And the problem is finding them, targeting them, and destroying them while not hurting the other cells. Okay, and we really haven't come up with the best solutions yet. I mean, there's chemo, and that hurts everything. Well, not everything. It hurts rapidly dividing cells. Chemo is going to attack all of your cells that are dividing rapidly. Let's say, we're talking about a cancer patient. The chemo will destroy the rapidly dividing cancer cells, but then it'll destroy the rapidly dividing normal cells too. So what are some rapidly dividing normal cells? Okay, your skin, your entire GI tract. That's why chemotherapy patients get sick and vomit. Your hair is made of proteins, and that is rapidly growing, and that's why people's hair falls out and things like that. All right, so if we had a universal, that's why we don't have a universal solution for cancer. We don't have one, because we would have to design a medication for every single human being that has the disease, and nobody has that kind of money. I mean, we're talking hundreds of millions of dollars to even get a specialized type of DNA. A specialized type of cancer medication for one individual. It's just not even possible right now. Maybe someday, I don't know. I mean, they don't put enough money into research, so that's another problem. All right, but if you've ever wondered, why can't we just get rid of this horrible disease? Well, that's why. That's one of the bumps in treating cancer. All right, now speaking of cancer, there's different types. Of course, like we all know this, there's benign and malignant. Ultimately, something that's benign is going to just not invade other tissue. It'll stay like this as a tumor. Okay, now if it starts to invade tissue, like these glands here, that's a problem. Okay, when it has fully metastasized, okay, it gets into your blood vessels and the lymphatic system. Okay, your blood vessels is your network that travels your entire body. So once it's in the blood, it can go anywhere. Now, there are patterns. They've found that it'll usually move to the next organ, but not always. It can get in there, and basically that cardiovascular system is going to be pumping those cancerous cells all over the body. All right, so that's enough of that. Cancer is sufficiently terrifying. Okay, we're going to move on to meiosis. Now, earlier I talked about mitosis, right? And it's for growth, development, cell renewal, cell repair. Now we're going to talk about meiosis, which is what? It has one purpose and one purpose only, and that is to make these guys, to make the haploid gamete sex cells. That's it. All right, so we're going to learn how that happens. Okay, so what we're looking at here is homologous chromosomes. So earlier I mentioned them. Now, the human genome has 23 pairs of homologous chromosomes. Homologous means the same shape, the same length, the same centromere position, and the same gene location. Okay, so a couple of terms that we have to know here is number one, homologous chromosomes. Okay, you as an individual, you have 23 pairs of homologous chromosomes, so a total of 46. And each one of those chromosomes is going to have the same type of genes, the same gene location. So another term we need to know is locus. Locus is the location or position of a gene. So this little yellow kind of mark right here, okay, these are genes. This green color, that's another gene. And the blue, all of these are representative of different genes along this chromosome. All right, so homologous chromosomes are the same chromosome with the same shape, length, and centromere position, but they are not identical. They are not identical because you get one from your mother and one from your father. So they are not identical. Okay, you inherit one from mom, one from dad, and they're going to be carrying the same the same genes. Of course, the genetic information will be different because your parents are different people. So earlier, you saw this human karyotype. Now it'll make a lot more sense. So for example, this right here, these are 2 chromosomes. Okay, this is homologous chromosome set number 1, homologous chromosome set number 2, homologous chromosome set number 3. All of these are the same size. Okay, see that? The same shape. This is one chromosome. This is the other next to it. Okay, these are the same size, shape, and they carry the same genes. Okay, and then you get one from your mother and one from your father. And of course, the last set are the sex chromosomes. And yeah, technically, the XX are completely homologous and the XY are not necessarily homologous because the Y chromosome is so different. But either way, it doesn't matter. The point is that when we say homologous, we mean what we said earlier. Same shape, size, same genes. But of course, you get one from your mother, one from your father. Okay, and that goes through 1 all the way to 23. Okay, so we technically have 22 pairs of autosomes and then 1 set of sex chromosomes, which we've already discussed. Now, we know that if a cell is going to enter the cell cycle, it's going to have to duplicate all of the chromosomes, right? So that happens in meiosis as well. So we're going to talk about meiosis. It is the whole purpose of meiosis is to produce gametes. But it's important to understand all of the steps, and not just that, we need to understand exactly what's going on. So let's go ahead and start. So meiosis is taking the chromosome, we're taking our chromosome set, our diploid set, and reducing it by half. So a human cell that's going to go through meiosis is going to start off as a diploid cell and end as a haploid cell. And this is how the gametes are produced. At the end of meiosis, this is a huge variance from mitosis. We're going to end up with 4 haploid cells. And they're going to be genetically unique. In meiosis, there are 2 rounds of cell division, and there's a process called crossing over, which leads to something called genetic diversity. All right, let's take a look. Okay, so this is a short, quick version of meiosis. And basically what they're emphasizing here is that there's 2 divisions. Okay, so basically, let's go ahead and kind of look at what they've done here. So they've given us this parent cell right here. Okay, and they've given us 1 set of homologous chromosomes. See how 1 is red? I'm going to zoom in, and 1 is blue. This is indicating that the blue one is the paternal one. So it's from the father. That's why they make it blue. Okay, the red one is from the mother. And we're not going to draw out 46 chromosomes were 23 pairs, is too much, right? So we work with no more than 6 at a time for learning and teaching. Okay, so here we have one set of homologous chromosomes, and you know they're homologous because they're the same size and shape, and their centromeres align. All right, now in this process, interphase is going to happen. And an interphase is in meiosis is just like the interphase in mitosis, meaning that there's going to be a G1, an S phase, and a G2. So all of the chromosomes are going to get duplicated, and they're also going to get paired. Okay, so here we are. Interphase has occurred. Okay, and if we look at this group now, we can see that everything has been, the maternal one has been duplicated, and so has the paternal chromosome. So if we count these, there's 1, 2, 3, 4 chromosomes when we started with 2. Okay, so we've doubled the DNA, and now these are called sister chromatids. Now you should remember that from mitosis. The terminology is similar. Okay, then the first split happens, called meiosis 1, and it's going to separate homologous chromosomes. Okay, that is a key factor. Meiosis 1 is the separation of homologous chromosomes. Okay, so that's what's happening in meiosis 1. Okay, so you see that the red's gone up here and the blue's gone over here, and then meiosis 2 is just a straight division. So there's not another duplication, there's no more copying. We're just going to cut the sister chromatids in half. And that's what meiosis 2 is noted for. Okay, so the key process that's going on here in meiosis 2 is that sister chromatids are separated. Okay, they're separated. Okay, so we can see that here, the separation of, this is one cell, it's going to split, and it's going to separate the sister chromatids into their own cells. Okay, this cell down here carrying the paternal chromosome, the sister chromatids will separate and get distributed into their own cells. So by the end of meiosis, we're going to end up with 4 cells. Now, if you remember from mitosis, how many cells do we end up with in mitosis? Mitosis, we end up with 2 genetically identical diploid cells. Here in meiosis, we end up with 4 genetically unique haploid cells. Okay, now this is the overview. Let's look at the details. All right, so this is pretty straightforward. You've seen these terms before. So notice this. Meiosis 1 is right here, and meiosis 2 is over here. And I'm going to go ahead and label that real quick. This is meiosis 1. Okay, this is meiosis 2. And I want you to notice that the terminology is the same. So as we see in mitosis. So prophase 1, metaphase 1, anaphase 1, telophase, and cytokinesis 1. Then we're going to see prophase 2 happening to 2 cells simultaneously, metaphase 2, anaphase 2, telophase, and cytokinesis 2. All right, so the terms are the same in regards to memorizing the stages, but of course there's 2 rounds of it. So we're going to look closely at meiosis 1 and 2, and then that'll be that. So the very beginning is the same as mitosis. Interphase. Every cell that's going through cell division is going to have to go through interphase. Okay, so that means G1, everything's going to be duplicated, the organelles, the cytoplasm, the plasma membrane. The S phase, all of the chromosomes are duplicated, and then G2. Now, all right. So interphase is the same, and then here's one of the primary differences. In prophase 1, meiosis 1, it's pretty similar except for a couple of things. Okay, it's similar in the sense that the mitotic spindle is going to form. There's a spindle right there. The nucleus is going to fragment and disintegrate. Now, there's two major events that occur that only happen in prophase 1, meiosis 1. The first thing that happens is called synapsis. Synapsis is when homologous chromosomes physically come in contact and pair up together. So this doesn't happen anywhere else except for here. So if you look really, really close, let's zoom in a little bit. Okay, the synapsis that we're talking about is, see this red X and the big one and the blue. Okay, these are the red here, of course, are sister chromatids. The blue are also sister chromatids. But notice this. See how these guys are physically close to each other? And see how these little arms are crossed over? Okay, that's not something that happens everywhere. See that? Look at this one down here. This is synapsis. Okay, so what happens here is all of the chromosomes, the homologous chromosomes. So dad's, the homologous chromosome number 1, the paternal, the dad one, will pair with the maternal one. And so will the second one, and third and fourth, all of them. All of the homologous chromosomes that we have are going to synapse, and they're going to physically become close to one another, and they're going to make something called a tetrad. Okay, a tetrad. Why would we call this a tetrad? Okay, a tetrad is, tetrad means 4. Okay, that's what tetra means. And if you count up these chromosomes, there's 1, 2, 3, 4 after the synapsis. Same thing down here. Count these up. 1, 2, 3, 4. 1, 2, 3, 4. Okay, so we don't see this phenomenon anywhere else except for prophase number 1, and this is called the synapsis. Now, what happens next is called crossing over. There's going to be a swap. Okay, see how these guys are physically in contact with each other? Right there, right here. Okay, throughout the tetrads, there's a physical contact between the blue and the red chromatid. Okay, they're going to swap genetic material. That is called crossing over. So what happens is, let me actually zoom in over here. All right, so you have your chromosomes, your homologous chromosomes. Of course, these are sister chromatids. Now together, the 4 are a tetrad, okay, and they're also referred to as a homologous chromosome set, even though there's been a duplication. These are still called homologous chromosomes. Okay, they're homologous because this is the paternal, this is the maternal. Okay, they're also referred to as the tetrad because there's 4 chromosomes. So this is synapsis. They synapse, they click together. Okay, then the next part is they're going to physically make contact, like right there. And then they're going to swap genetic material. That is called the crossover or crossing over. And now you see that there's been a swap. A little bit of red is at the end of the blue and vice versa. Every homologous chromosome set is going to do this. And this is the reason why genetic crossing over is the reason why you are completely unique. Why every single human being on the planet is completely unique other than identical twins. And it's because every single time the genetic or the gametes are produced, the sperm and egg are produced, there's a crossing over. There's a genetic shuffle. So every egg and sperm that's made is going to be slightly different. There's always going to be a genetic crossover, and we don't know exactly how that's going to turn out until there's a person, a human that's born, or an animal, or whatever. Let's just stick with humans here. Okay, so basically, this is incredibly significant because it provides us with all of the genetic diversity that we have in this world. I mean, this is why you're not a clone of your parents. This is why you don't look exactly like your siblings if you have them. And this is what makes everyone unique. Everyone. So it's kind of interesting in that sense. All right, I think we get it. So crossing over, just so we're clear, synapsis and crossing over happen in prophase 1, meiosis 1. Okay, metaphase 1. This is incredibly important. Homologous chromosomes, tetrads, they're going to align at the metaphase plate. All right, now if you look closely, notice that there's 1, 2, 3, 4 homologous chromosomes. The tetrads are aligned at the metaphase plate. Unlike mitosis, where sister chromatids align at the metaphase plate, this is completely different. So we've done the synapsis and crossing over. That's all done. So the cell's ready to roll. However, we need to pull these homologous chromosomes apart. And the first way to do it is by getting them set up at the metaphase plate and by pulling them apart. And of course, that's very easy to do once we have the mitotic spindle attached to the centromere of each sister chromatid set. Okay, so in anaphase 1, meiosis 1, what's going to happen is homologous chromosomes are pulled apart. So see these little guys? Okay, and they have, you can see the little crossover right there on each one. All right, so, but this is important. Sister chromatids stay attached. What's been separated? Homologous chromosomes. Remember the terminology. It's going to be key. Now, telophase and cytokinesis, I really don't have to go through it because it's straightforward. What's going to happen is the same telophase is going to occur that occurs inside of mitosis. So meaning that the nucleus will regenerate. It's going to form around these cells. I'm sorry, not cells, these chromosomes. Okay, the mitotic spindle will go away, and then cytokinesis will cut the cell in half, and we're going to end up with 2 cells. Okay, each cell is going to be carrying 46 chromosomes. Now we're going to move into meiosis 2. Meiosis 2 is pretty straightforward. The whole purpose of this is to pull apart the sister chromatids. All right, now we've already gone through meiosis 1, and we split that cell, and we have 2 cells, okay, and they're going to go through meiosis 2. So they'll go through prophase 2, metaphase 2, anaphase, and telophase and cytokinesis 2. So how does that work? Straightforward. It's actually exactly like mitosis in the sense that here we are, we got our mitotic spindle's going to form, and on top of that, the nucleus is going to disintegrate again, and these mitotic spindle arms are going to grab onto the kinetochore of sister chromatids. And what is aligned at the equator? Sister chromatids are aligned at the equator. Okay, and that's for both cells. Anaphase 2, sister chromatids are separated. Telophase and cytokinesis, we're going to end up with 4 cells. And they're going to be haploid because there's 1, 2, and they're going to be genetically unique. Okay, so if we're cutting this in half and then this in half, we're going to end up with 4 haploid cells, right? And they're genetically unique. Why? Why are they genetically unique? There's only one answer to that. They are genetically unique because of the synapsis and crossing over from meiosis 1. Okay, so that's how meiosis happens. We're going to watch a quick video animation on that. I want you to take notes, and then we will wrap up this lecture.
Many organisms pass their genes to their offspring through sexual reproduction. This begins when 2 gametes unite to form an embryo that is genetically unique from the parent organisms. The embryo then grows into an adult who in turn passes their genetic information on to their own offspring. Gametes are formed through a process called meiosis. The cells that undergo meiosis to produce the gametes are called germline cells. In diploid organisms, germline cells have 2 copies of each chromosome. Germline cells undergo meiosis to produce haploid gametes, which only have 1 copy of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into the adult. Meiosis is just one step in the life cycle of a germline cell. Similar to mitosis, the cells also pass through the interphase, G1, S, and G2 stages before they enter meiosis. The DNA inside a germline cell is duplicated before meiosis begins during the S phase. The duplicated germline chromosomes are called sister chromatids. These chromatids remain attached to each other until the second cell division event in meiosis. There are 2 cell division events during meiosis. The first division, meiosis 1, results in 2 unique daughter cells that have half the amount of DNA as the parent germline cell. The second division, meiosis 2, results in 4 unique haploid cells that only have 1 copy of each chromosome. These haploid cells are the gametes that could go on to produce an offspring through sexual reproduction. Let's look more closely at each of the division events. Meiosis begins with prophase 1. In this stage, the DNA condenses to form chromosomes. Here we see the duplicated sister chromatids joined together at the centromere. They stay fused at the centromere throughout meiosis 1. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving 2 pairs of sister chromatids. Chromosomal material is exchanged between the 2 pairs of sister chromatids. This event is called recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no 2 siblings, aside from twins, are genetically identical. There are several more key steps in prophase 1. The nuclear membrane begins to break down. Then, the 2 centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis 1 is called metaphase 1. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is anaphase 1. During this phase, homologous chromosomes separate and migrate to the two poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis I are telophase I and cytokinesis. Here, the cell divides into two daughter cells. Each of these two cells now undergo meiosis II. Meiosis II is similar to mitosis. The first stage of meiosis II is prophase II. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase II and prophase I is the fact that the daughter cells have only one copy of each homologous chromosome. So in prophase II, there is no synapsis of homologous chromosomes or crossing over. In metaphase II, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align. In anaphase II, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In telophase II, the nuclear membrane reforms and the cytoplasm is divided into the two haploid daughter cells. This division is called cytokinesis. Since meiosis II began with two cells, and each of those cells were split into two cells, we now have four unique haploid cells at the end of meiosis. These cells are gametes. Two gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis.
Okay, so at this point, you guys should have watched all of the cell division lectures that I have on YouTube. Now, what I have here is what I would normally draw on the board for my class. However, since we are going online, it is important to make sure that you have all of the materials that you would have whether you were in the class or not, and this is part of it. And I would like for everyone to take this diagram and redraw it. I know you have a copy. I've given you one under assignments, and I would like for you guys to actually redraw the entire mitosis and meiosis process, because that is how you will learn this material, become an expert at this material, and succeed on your next exam. Okay, so let's start. Now, we know that in mitosis, mitosis just is this small piece of the pie, right? Or a piece of a big cycle, which is the cell cycle. Now, we know the stages of mitosis, and now before we get to that, let's talk about what we have here. So every time mitosis happens, it happens with a cell, a parent cell, we call it, and it's diploid. In this case, the 2N diploid number is 4. We're choosing to use 4 chromosomes to teach with rather than 46, which would be overwhelming. Okay, so ultimately, if we're talking about human cells, and the number count is different, but when we teach with the 4 cells. All right, so here we go. What's going on here is I've created 4 cells. 1, 2, 3, 4. Okay, these 2 are long and they have the same letter, except one is capital, one is lowercase. Capital A, lowercase a. This is indicating they're the same size, shape, and they have the same information, aka the same letters. Therefore, they are homologous chromosomes. They are a pair. Okay, this is another pair of homologous chromosomes that are short, a little shorter, and then they're the same size, shape, and then they have the same genetic information using the letter B. Now, when interphase happens, we know the cell is going to grow, duplicate all of the organelles, plus it's going to duplicate DNA, meaning all of the chromosomes in the genome. And that happens during which phase? Now, if you remember correctly, the synthesis of DNA and the duplication of chromosomes happens in the S phase. So when we enter prophase in mitosis, here we are going to have sister chromatids. There's the original, the duplicate. Pay close attention to my labels. Notice that in the center, what's holding them together is a centromere. And on top of that, these are now called sister chromatids. Sister chromatids, sister chromatids, sister chromatids, sister chromatids. Notice that each sister chromatid has another protein called the kinetochore. The kinetochore. Okay, and these are identical. If you look at them, they are identical. Same capital letters, same length, shape, same thing here, here, and here. All right, so we've duplicated everything. So if we were to give a chromosome count, everything's been doubled. We now have 8 total chromosomes. You can count them up. Okay, the mitotic spindle is forming or has formed. These are the centrioles. These mitotic spindles can grow, and they're going to move to opposite poles of the cell, and they're going to connect to the kinetochore. This set will connect to this kinetochore of that sister chromatid, and this one will connect to the other. And it'll happen for each and every set of sister chromatids. The nuclear membrane will disintegrate, and meanwhile, we don't exactly see it unless we're watching an animation. By the time we get to metaphase, these microtubules of the mitotic spindle have they've already connected to all of the kinetochores, and they've pushed and pulled and aligned and realigned, and they have placed or aligned sister chromatids on the equator of the cell. Now they're ready to get pulled apart, and that's what happens in anaphase. Anaphase is the separation of sister chromatids, and that happens by the shortening of the microtubules of the mitotic spindle. Okay, and then we have telophase. We have our cleavage furrow. The nuclear membrane is going to return, and it's going to come back, and notice that all of these chromosomes on top will end up here. All of these chromosomes on bottom will end up down here. And we will get our cleavage furrow, and this will continue to cleave through cytokinesis until we get 2 genetically identical daughter cells, and they are genetically identical. Okay, and that is mitosis. Meiosis, remember, is quite different, but very similar. We're going to start, it starts with the same parent cell, okay, no different. And same thing in the human body and in any body, it always starts off with the full genome, and then interphase is going to occur. However, what's different here is the synapsis of the homologous chromosomes. Now, these are sister chromatids, so are these. Okay, but now they synapse, and they physically synapse. These guys will physically synapse. Everything else is the same as mitosis, like the nuclear membrane fragmenting, mitotic spindle occurs shows up and develops. And these centrioles are actually going to connect, sorry, not the centrioles, the mitotic spindle is actually going to connect to the centromere of each and every sister chromatid. It's going to push, pull, realign, and there's the crossovers happened. And so when we get to metaphase, what we have on the equator are homologous chromosomes, tetrads. Okay, 1, 2, 3, 4. And notice the marking in red and blue indicating that there's been a genetic crossover. Now, very quickly, what is a line at the metaphase plate in mitosis? Sister chromatids. That is so important because that is how you are going to answer questions and how you are going to navigate between the two. You have to know these terms. You have to. Okay, that's why I want you to draw them. All right, so here we go. Metaphase 1, homologous chromosomes align at the equator. Anaphase 1, it's the separation of homologous chromosomes. And here we go. They have been separated. And then we have a telophase and cytokinesis, and we're going to end up, this is meiosis 1. We end meiosis 1 with 2 diploid cells, okay? And then meiosis 2 is going to happen simultaneously. Prophase 2 is going to be the same as prophase. Nuclear membrane disintegrates, mitotic spindle shows up, happening on both cells, and then metaphase 2. How we distinguish it is because sister chromatids are aligned at the equator, very similar to mitosis. Okay, anaphase 2, sister chromatids are separated. Telophase and cytokinesis 2 is just simply the fact that the cell will reform its nuclear membrane around the chromosome. The chromosome uncoils into chromatin. Mitotic spindle goes away. This will get cut in half. This will get cut in half. And we have 1, 2, 3, 4 genetically unique daughter cells. Now look, are any of them identical? This one, these two are not, because look at these markings. That makes them unidentical. Here, same thing. Marking, not identical. Okay, so that is mitosis and meiosis. I want you to draw this animation. You can use it for your cell division lab to draw, but you have to know every detail. And the best way to learn every detail is to draw every detail. Okay, this slide is a quick compare and contrast of mitosis and meiosis. I think we've made several comparisons, but this is a good review for you guys to do on your own. So obviously this is the mitosis column, and this is the meiosis column. And the point is to compare the similarities and differences. Of course, there are similarities, and then primary differences. The primary differences are the synapsis and crossing over in prophase 1, the fact that there's 2 divisions inside of meiosis, and we end up with 4 genetically unique haploid daughter cells in meiosis, and at the end of mitosis we end up with 4 genetically identical diploid daughter cells. All right, now ultimately, we talked about how meiosis is the formation of the gamete sex cells, which is true. Now, once the meiosis has occurred, there has to be further maturation of the cells. Okay, so that is where spermatogenesis and oogenesis comes in. Okay, spermatogenesis is clearly when the sperm is being formed. A cell, that person's cells with the full set of chromosomes will go into meiosis 1, split. Okay, it'll follow all of the rules of meiosis. And then it'll go through meiosis 2, giving us 4 genetically unique cells. All right, but those are not fully sperm cells, right? So they have to go through a maturation, and then we end up with the 4 sperm that are genetically unique, carrying 23 chromosomes apiece. Now, oogenesis is a little different. So the beginning is that individual's diploid cell count. And then, of course, it will go through the whole process of meiosis 1, interphase, then meiosis 1. And notice this, when meiosis 1 occurs, you're going to see the chromosomes will be separated. However, what's going to happen is the majority of the cytoplasm is going to lean towards one side of the cell. So when division occurs in the first round of meiosis, you'll get one cell, and then you'll get this little kind of unit of chromosomes, and it's called the polar body. Okay, then when meiosis 2 happens, you'll see the cytoplasm will lean over to one side, and basically the same thing will happen. The sister chromatids will be split up. The cell will technically divide, yes, but if the cytoplasm and plasma membrane lean over to one side, then we end up with one cell and another polar body, which is fine as long as the chromosomes are separated. And in the end of meiosis 2, and in the end of oogenesis, which is the maturation of an egg, we end up with one egg, and this is per one ovary per single month. Okay, so what happens to a body or an individual or a situation if meiosis goes awry? Now, meiosis should occur perfectly in every single part of meiosis 1 and meiosis 2. If it does not, there are severe consequences. Severe. All right, so this is what happens. 2 possible problems. Number 1. Okay, first of all, this whole thing that I'm about to discuss is called non-disjunction. Non-disjunction is the failure of chromosomes to separate. Okay, so non-disjunction can happen at meiosis 1. Here you can see that these chromosomes have separated the way they're supposed to, right? And these have not. That is not okay. That is non-disjunction. And why is it not okay? This is why. All right, so let's say that happens, and then we go into meiosis 2. Okay, well, that's going to cause an unequal distribution of chromosomes. When we go into the last division, we are going to have abnormal cell count, abnormal chromosome count. See how this says n+1? It's because there's 1, 2, 3. There's +1 chromosomes. There should be 2 there, but there's not. There's 3. Here, there's also 3. Here, there's 1 missing, and there's 1 missing in this one. They're all abnormal, okay, because the non-disjunction threw off all of the cells. That means that these gametes, all 4 of them, are carrying an incorrect number of chromosomes. Now, if this egg, let's say this is an egg, okay, if this egg goes on and it gets fertilized and becomes a baby, that baby is going to have a chromosome disorder because of meiosis, having an error, the non-disjunction. So everything has to go perfectly, or we can end up with these catastrophic results. Okay, let's look at the second type of non-disjunction. So let's say meiosis 1 goes perfectly fine, normal, no problem. Okay, great. And therefore, this cell will split, and on top of that, all of these guys will be fine, and the non-disjunction could potentially happen in meiosis 2. Now, is it likely for it to happen in both cells? Not really, no. That's pretty unlikely. So usually it'll happen in one cell. Okay, so it's the failure of sister chromatids to separate. And so in the end, we get 4 cells, right? These are normal, 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 2 of 4 of normal, and then these are abnormal. So we're going to have abnormal gametes. Now, if a couple is having a child and this egg gets fertilized, or that sperm fertilizes an egg with an unequal distribution of chromosomes, or basically the incorrect amount of chromosomes for a human, and that becomes a child, that child is going to have a chromosome disorder. And of course, no one's going to know until the baby's in utero. All right, so that is the critical the importance of meiosis. Now, earlier when I talked about mitosis, I said that it had to be perfect, because if it's not perfect, then that could lead to cancer, right? And now here we're talking about meiosis. Now, meiosis has to be perfect, and if it's not, the consequences are a chromosome disorder. Okay, now let's just kind of recap here. Non-disjunction is either the failure of homologous chromosomes to separate or the failure of sister chromosomes to separate, one or the other. Okay, and ultimately it's going to, if this occurs, and if one of these cells that had non-disjunction occur in the process goes on and gets fertilized, we could end up with some major diseases, like, for example, Down syndrome. Down syndrome is called trisomy 21. What does that mean? All right, Tri. Okay, this is important. Tri equals 3. Okay, and then 21 means location 21. Okay, let's go and look at that. All right, here we go. This is Down syndrome. So remember the human karyotype. Zoom in on this. Okay, remember the human karyotype, we're supposed to have 23 pairs of chromosomes, right? 22 autosomes, 1 pair of sex chromosomes, and we're supposed, pair means 2, of course. And so here, everywhere, we have 1, 2, 1, 2, 1, 2, at each location, 1 through 22. Okay, now people with Down syndrome, they have a 3rd chromosome at location 21 on the karyotype. That is why this is called trisomy 21. This is Down syndrome. So this is caused by meiotic error. This is caused from non-disjunction, meiotic error. So there's other trisomies. There's trisomy 13, there's trisomy 18. This is not the only trisomy. In fact, there are trisomies that alter the sex chromosomes. Okay, like Jacob syndrome is an interesting one. It's an XY. So it's a male, right? But they have an extra Y chromosome. And usually they don't even know that they have it, actually. A lot of people with Jacob syndrome can go on their whole life and not even realize that they have Jacob syndrome unless they get a full genetic panel. Mainly the thing with people with Jacob syndrome is because they have the extra Y chromosome, they're going to have extra testosterone, which is they're going to be taller. They might be a little bit more aggressive. They might have some learning disabilities. It just depends on the individual. But for the most part, they can lead a relatively normal life. Okay, there's also Klinefelter syndrome. So this is an XXY. So it's a male that has an extra X chromosome. So they're going to have extra female properties. So I'll show you a picture really quick. Klinefelter syndrome. Okay, so they're showing many X's. Really, it can be that messed up, but it shouldn't be. It should be XXY. So there should be two X's and a Y. And typically, this is going to have a drastic effect on the individual. Their life's a little difficult. They're going to have less muscle mass. They'll have kind of some female features like hips and breast tissue, and their bone structure will be weaker. And usually, they are sterile. So that's a tough life, and that's a chromosome disorder. This one is too. This is called Turner syndrome, and this is one of the rare ones that's blank. It's got an X and then nothing. There's no other X, there's no Y, so it ends up being a female, but there's a lot of problems there. They stay very short like that. They have no neck. Some of them have webbed feet, and it can be very, very, very difficult. All right, so all of this is caused from meiotic error. Everything from Down syndrome to Klinefelter to the Turner syndrome. Okay, so either way, if meiosis does not happen perfectly, and individuals end up, reproducing, they can have children with various genetic disorders, and we won't know what it is until, at least 2 months into the pregnancy, they could start doing genetic testing. Okay, so for example, this is another one that's kind of interesting. The other one is called trisomy 13. Okay, so that means that there would be a third chromosome here. So trisomy 13 is a chromosome disorder. And basically, there's a point in the pregnancy where the doctor will do a genetic panel and look for Down syndrome. They'll look for any chromosome disorder. It's pretty simple these days. They can do a quick test, but it does cost money. That's an issue, but most people get it done. And trisomy 13 is a terrible disease. Basically what happens with that is the babies if they make it, they will be born with parts of their brain missing, their eyes missing, and they don't live past a year. That's trisomy 13. So a lot of the time, when they discover that, they're advised to not continue with the pregnancy, and it's really up to them. That's going to, be something that becomes an ethical, moral question that every, individual in that situation will face. But is it, there's also the question of, is it right to bring a child with missing part of their brain and eyes and let them live for 6 months to a year and then die? It's just kind of one of those situations that you kind of have to weigh the pros and cons, and every person makes their own decision privately. But those are the kind of things that come up in life and in general. All right, so if you ever hear about a genetic disorder like that, this is where it comes from. This is the origin. It's a mistake in meiosis called non-disjunction. Okay, now I want you guys to carefully review this chapter and make sure you diagram mitosis and meiosis and know them inside and out. And that is the end of chapter 8.